All right, so let's dive right into this deep dive, all about mutagenic impurities and ICH-M7 guidelines.
Yeah.
You sent over a ton of material, actually. I mean, from regulatory documents to like scientific studies and even some pretty interesting real-world examples. So it seems like you're really trying to understand how we ensure that the pills we take are safe, especially when it comes to like, those potential cancer risks.
Absolutely. Yeah. It's a complex area for sure.
For sure, yeah.
ICH-M7 is all about identifying and controlling, well, you could think of them as these sneaky little molecules that could potentially mess with their DNA and cause all sorts of problems down the line.
Okay, so we're talking about DNA reactive impurities, right?
Right.
But how do scientists even know which impurities are the troublemakers? And then, like, how much is too much?
Yeah, well, that's where the concept of risk assessment comes in. You see, ICH-M7 isn't about just like eliminating every single impurity out there.
Right.
It's about figuring out the level of risk each one poses, and then, putting in the right controls. It's a real delicate balance between, making sure things are safe,
right,
but also allowing for development of new and effective medications.
So how do they actually assess that risk? I imagine it involves a lot of serious science going on.
Oh, it does, yeah. One of the key tools that they use is something called QSAR.
Okay.
Quantitative structure-activity relationship.
Wow.
It's essentially software that can predict, how likely a molecule is to be mutagenic based on its structure. So think of it as this, I guess, a virtual detective, for identifying these potential bad actors.
That's really fascinating. So they're basically like analyzing the molecular fingerprint to try to predict how it's going to behave.
Yeah, that's a great way to put it.
Okay, so what happens once they flag these potential mutagenic impurities? Like, what's next?
Well, then they need to figure out how much of that impurity is actually acceptable. That's where, that's where the concept of acceptable daily intake, or ADI, comes in.
Okay.
And this is the amount of a substance that a person can be exposed to every day over a lifetime without any noticeable risk.
Okay, so it's like setting a safety threshold.
Yeah.
But wouldn't that threshold vary depending on, the drug and how it's used?
Yeah.
A short course of antibiotics is different from a daily medication you take for years.
You're exactly right. The ADI is calculated differently for chronic treatments,
Okay.
like, for high blood pressure,
Right.
versus like acute treatments, like a course of antibiotics.
Yeah.
For chronic treatments, the ADI is set much, much lower because that exposure is over a longer period of time.
Okay, that makes sense. But how do they actually determine these ADIs? Is it just like based on scientific intuition, or is there like a more rigorous process?
Oh, no. It's definitely not based on guesswork.
Okay.
Scientists use pretty sophisticated statistical methods to analyze data from all sorts of studies.
Mm-hmm.
And it's ensuring a very high degree of confidence in those calculations. Think of it like, you're making sure your measuring tools are perfectly calibrated before you start baking.
Okay, so there's a lot of statistical validation going on there.
Right.
What I find interesting is that ICH-M7 seems to impact the entire drug development process, not just, like the final product.
Absolutely. It starts with those really early preclinical studies.
Yeah.
Where they're testing the drug in a lab setting, often using animal models to gather that initial, safety and effectiveness data.
Right.
And then as the drug moves into clinical trials with human participants, ICH-M7 considerations continue to guide the process.
And speaking of clinical trials, I remember from the materials you shared that the FDA plays a big role in overseeing all those trials, right?
Yes, the FDA's role is crucial there.
Yeah.
They, they review the trial design, they monitor the data, and they ultimately decide whether the drug is safe and effective enough to be approved.
Okay.
So they're kind of like, like the quality control experts, making sure everything meets the highest standards.
There's not just scientists in a lab.
Right.
There's this whole regulatory system ensuring patient safety.
Exactly.
Now, you mentioned that ICH-M7 influences how those mutagenic impurities are controlled throughout that whole process.
Yeah.
Can you elaborate on that a little bit?
Sure. One crucial aspect is the development of really robust analytical methods.
Okay.
So scientists need incredibly precise tools to detect and quantify those impurities even when they're present in tiny amounts. It's like having this, a super-powered microscope that can spot those microscopic troublemakers.
And I bet that gets even more challenging when you're dealing with, like, complex manufacturing processes, right?
Oh, you're right. Yeah. Each step in the manufacturing process has the potential to either introduce or remove impurities. And that's where the concept of purge factors comes in.
Purge factors, yeah.
These factors, well, they actually measure how much of a specific impurity is removed during each step.
Oh, okay.
So it's like, having multiple layers of filtration,
Right.
each one designed to kind of remove those unwanted impurities.
But how do they ensure that this whole intricate system is actually working as intended?
That's where meticulous documentation comes in.
Right.
Remember that? Remember the Common Technical Document, or CTD?
Yes, the CTD. From what I understand, it's a really comprehensive record-keeping system.
Mm.
It kind of tracks the drug's entire journey from its initial development all the way to its final approval.
Yeah, exactly. It's like a detailed logbook.
Right.
That ensures traceability and accountability.
Okay.
And within the CTD, there's a very specific section that's actually dedicated to information about mutagenic impurities.
So it's not just about, following the rules. It's about documenting every single step of the way to really demonstrate that compliance.
Right.
It seems like ICHM-7 and the CTD work hand in hand to create, a pretty robust system for ensuring drug safety.
They definitely do. And in the next part of our, deep dive, we'll delve a little deeper into how these guidelines are actually applied in these real-world scenarios, and explore some specific examples from drug stability testing to the development of new drug substances.
That sounds fascinating. I'm eager to hear more about how all those pieces actually come together in practice.
Stay tuned.
Yes.
Welcome back to our deep dive into the world of mutagenic impurities and ICHM-7. We've already laid some groundwork, right? Exploring those core concepts of risk assessment, ADI calculations, and of course, the role of QSAR in identifying those potential culprits.
And we talked about how ICHM-7 guidelines influence everything from, those early preclinical studies to those pretty rigorous clinical trials overseen by the FDA. It's amazing how much actually goes into making sure those pills we take are safe.
Oh, it's a complex process for sure. But it's all about making sure those medications are both effective and safe for patients. So let's shift our focus a bit to the manufacturing process itself.
Okay.
A crucial stage, right, where those purge factors we discussed earlier really come into play.
Right. Those multi-stage purification processes designed to remove all those unwanted impurities at each step. It's like a series of carefully designed filters,
Exactly.
each one refining the drug substance a little bit further.
Exactly. And to ensure those manufacturing processes are really up to par, there's a whole set of regulations known as good manufacturing practices, or GMPs. And these guidelines are like this strict recipe, right, that manufacturers have to follow, ensuring the quality and safety of their products.
GMPs. I've heard that term before, but can you break it down for me a little bit?
Sure.
How do they actually relate to ICHM-7 and the control of mutagenic impurities?
Well, think of GMPs as the overarching framework for quality control in pharmaceutical manufacturing. They cover everything from facility design and equipment maintenance to personnel training and documentation procedures.
So GMPs are kind of like the foundation upon which ICHM-7 is built.
Precisely. ICHM-7 is it's specifically focused on mutagenic impurities, but it operates within this broader context of GMPs.
Got it.
So by controlling those impurities throughout the whole manufacturing process, companies can actually demonstrate their adherence to these GMP principles and ultimately ensure the quality and safety of that final product.
Okay. So we've got ICHM-7 addressing mutagenic impurities specifically, right?
Right.
And GMPs acting as this broader quality control framework. It sounds like a pretty intricate dance.
It is.
Between the science, the regulation, and the manufacturing.
It certainly is. But there's another crucial player in this dance that we can't forget.
Okay.
Documentation. Remember the Common Technical Document, the CTD?
Yes, the CTD. That really detailed record-keeping system that tracks the drug's entire journey. Like a detective's case file.
Exactly. And GMPs are essential for ensuring that the CTD is complete and accurate.
Okay.
Every step of the manufacturing process, from the raw materials that are used to, even the cleaning procedures for the equipment.
Right.
All of that has to be meticulously documented.
So it's not just about following the recipe. It's about keeping this a detailed record of every ingredient at every step along the way.
That's right.
I'm really starting to see how, how these pieces fit together. ICH-M7, GMPs, and the CPD, they all work in concert to create, this robust system
They do.
for ensuring the safety of, of medications.
They certainly do. And here's where it gets even more interesting.
Okay.
Remember those preclinical studies we talked about?
Yes.
Well, GMP principles actually extend to those, those early stages of drug development as well.
Really? I thought GMPs were, I thought they were mainly focused on, the manufacturing of the final product.
Right.
Why would they be relevant in preclinical research?
Well, while GMPs are, they're most critical during that commercial manufacturing phase.
Yeah.
They also play a role in preclinical studies because the data generated in those early stages can actually influence the design of that manufacturing process later on. It's about building a culture of quality from the very beginning.
So it's like laying the foundation for GMP compliance right from the start.
Exactly.
But adhering to these principles early on, companies kind of set themselves up for success.
Right.
And ensure the quality and safety of their product down the line.
Exactly. It's a very proactive approach that helps to minimize potential problems and ensures consistency throughout the entire drug development process. It's all about creating a seamless transition from the lab to the manufacturing plant.
I see. So GMPs are applied, from the very beginning.
Right.
And that meticulous documentation feeds into the CTD, right?
Yes.
Ensuring that traceability and accountability. This all makes sense. But I'm still curious about how those purge factors work in practice. Could you give me like a specific example?
Sure. Let's say you're developing a new drug substance, right?
Okay.
During that synthesis process, there's a potential for a certain mutagenic impurity to form.
Okay.
So to control this, you might introduce a very specific purification step, like crystallization, for example. That's designed to remove that particular impurity.
Okay, so crystallization acts like a filter, removing the unwanted impurity from that drug substance.
Precisely. The purge factor for that crystallization step would then be calculated based on how much of that specific impurity was removed.
Okay.
So a higher purge factor, that indicates a more effective purification step.
That makes sense. So they're essentially quantifying the effectiveness of each purification step.
Right.
But what happens if a particular step isn't as effective as they'd like it to be?
Well, that's where the iterative nature of drug development really comes in.
Okay.
They might need to go back to the drawing board,
Right.
and optimize that step. Perhaps they need to, adjust the temperature or the solvent.
Right.
Or even the duration of that crystallization process to improve its effectiveness.
So it's a process of continuous improvement, really. Always striving to refine those purification steps and minimize the risk of those mutagenic impurities making their way into the final product.
You got it. And this meticulous attention to detail is documented every step of the way,
right,
providing this clear trail of evidence for those regulatory agencies like the FDA.
This is all starting to make a lot more sense now. But I'm still curious about one thing. You mentioned that GMP principles extend to preclinical studies as well.
Right.
Could you give me an example of how that plays out in practice?
Certainly. So imagine you're conducting a preclinical study to evaluate the safety of a new drug candidate.
Mm-hmm.
GMP principles would dictate that you use well-characterized cell lines or animal models,
Okay.
ensuring that your results are reliable and reproducible.
So it's about, controlling for any potential variables that could skew those results, right?
Yeah.
Just like you would, in a manufacturing setting.
Exactly. And it extends to, the handling and storage of that drug substance as well.
Right.
You would need to follow very strict protocols, right, to prevent any contamination or degradation.
Okay.
Ensuring that the material used in the study is, it's representative of what would eventually be used in that final product.
It's fascinating how this attention to quality extends to every stage of that process. I'm starting to see how these, seemingly small details can have a big impact on the overall, safety and effectiveness of the drug.
They absolutely do. And that's why GMP compliance is so crucial. It's
Yeah.
about creating this culture of quality that permeates every aspect of drug development, ensuring that patient safety is always, it's always the top priority.
Well said. Now, I know we've been, focusing a lot on the technical aspects of ICH-M7 and GMPs,
Mhm.
but I'm also curious about that human element.
Yeah.
Are there any, ethical considerations that come into play when we're talking about, mutagenic impurities?
That's an important question. While we haven't dived into specific ethical dilemmas today,
Yeah.
it is crucial to remember that ICH-M7 is ultimately about protecting patients.
Right.
These guidelines are designed to minimize those potential risks
Right.
and ensure that the medications people rely on are as safe as possible.
That's a good reminder that, at the end of the day, it's all about ensuring that those medications are both effective and safe for the people who need them.
Absolutely, and that's why these seemingly complex, regulations and guidelines are
Mm.
so important. They provide a framework for making informed decisions. Balancing the potential benefits, with those potential
It's
risks.
like a delicate balancing act,
It is.
but it's one that's absolutely essential for public health.
Precisely. And in the final part of our deep dive, we'll explore some real-world examples of how these guidelines are applied in practice, from stability testing to drug interactions. So, stay tuned for even more insights into this fascinating world of pharmaceutical safety.
Okay, so we're back for the final part of our deep dive into mutagenic impurities and ICH-M7 guidelines. And we've covered a lot of ground, from the nitty-gritty of risk assessment and ADI calculations to the broader context of GMPs and the importance of meticulous documentation.
Yeah, we've really explored how ICH-M7 impacts that entire drug development process, from those early preclinical studies all the way to to that complex world of manufacturing,
Right.
ensuring that those medications we rely on are both effective and safe.
And I really appreciate how you've emphasized the interconnectedness of it all, the ICH-M7 GMPs, the CTD.
Yeah.
They all work together to create this really robust system for ensuring patient safety.
They do.
But I think what would really help solidify my understanding is seeing how these guidelines are actually applied,
Yeah. Yeah.
in real world situations.
Absolutely. Sometimes those regulations and guidelines can feel a bit abstract.
Yeah.
Until you see them in action. So let's dive into some specific examples.
Okay.
Starting with drug stability testing. Remember how we talked about the need to ensure those medications, remain safe and effective over time?
Yes, we touched on how drugs can degrade over time,
Yeah.
potentially forming, harmful byproducts.
Right.
And that's where stability testing comes in.
Exactly.
To make sure that they stay potent and don't pose any new risks.
Exactly. And ICH-M7 considerations are actually woven into those stability testing protocols.
Okay.
So, for instance, companies need to demonstrate that their manufacturing processes effectively remove or control any potential mutagenic impurities that might pop up during storage.
So it's not just about the initial purity of the drug. It's about making sure it stays that way throughout its entire shelf life.
Precisely. And those meticulous records we discussed, they come into play here as well.
All right.
Companies need to maintain very detailed records of their stability testing results.
Okay.
To demonstrate compliance with ICH-M7 and other regulations, it's like having this long-term monitoring system,
Right.
constantly checking in on the drug's safety.
I see. It's like having this built-in safety net to make sure nothing slips through the cracks.
Right.
And do these stability testing requirements extend to different, formulations of a drug? Like, for example, if a company wants to develop a new tablet form of an existing medication,
Yeah.
where they need to run, new tests.
Absolutely. Even seemingly minor changes to a medication, like a new formulation, can trigger a whole new round of testing. They need to ensure that the new tablet form doesn't, introduce any new risks or lead to the formation of, new mutagenic impurities.
That's fascinating. So it's not, it's not a one and done kind of deal.
Right.
It's this ongoing process of evaluation and monitoring.
Exactly.
Now, what about the development of entirely new drug substances? How does ICH-M7 come into play at that early stage?
It plays a crucial role right from the start. When scientists are developing a new drug substance, they need to assess its potential to form mutagenic impurities
Right.
during the synthesis or manufacturing process.
Right.
And this involves a combination of scientific knowledge, data analysis, and risk assessment tools to identify any potential red flags.
So it's like a proactive risk assessment, trying to anticipate and address any potential problems before they even arise.
Exactly. And if they do identify a potential risk,
Right.
they need to develop strategies to mitigate it. This might involve, modifying the synthesis route,
Right.
adding additional purification steps, or even selecting a completely different starting material. They have a whole toolkit of strategies to, address these challenges.
It's like they're playing a game of molecular chess, really, strategically planning each move to outmaneuver those potential mutagenic impurities.
That's a great analogy.
And of course, GMP principles are also critical at this stage, right?
Absolutely. They need to ensure that their manufacturing processes for those new drug substances, adhere to those very strict quality standards right from the get-go.
Right.
Minimizing the risk of contamination or the formation of unwanted impurities.
It really is a fascinating web of interconnected processes, all working together to ensure that those medications are as safe as possible. Now, I know we touched on this earlier, but I'm curious to explore the concept of drug interactions a little bit further.
Yeah. Oh.
How does ICH-M7 play a role in assessing those?
That's another crucial area where ICH-M7 guidelines come into play. When evaluating potential drug interactions, companies need to consider whether whether that combination of medications could increase the risk of forming mutagenic impurities, or even enhance their effects. It's like adding another layer of complexity to the puzzle.
So they're not just looking at how those drugs interact with each other in terms of their therapeutic effects, but also how they might interact in terms of forming those potentially harmful impurities.
You got it. It's a complex and challenging area of research, but it's absolutely vital for ensuring patient safety. After all, many people take multiple medications, and understanding these interactions is really crucial for minimizing those potential risks.
It's like this delicate balancing act, trying to optimize those therapeutic benefits.
It is.
While minimizing any potential harm.
Precisely. And ICH-M7 provides a framework for navigating that balance, guiding researchers and manufacturers in making informed decisions, throughout that entire drug development process.
Well, I have to say, this deep dive has been incredibly insightful. I feel like I've gained a whole new appreciation for this complex world of pharmaceutical safety and the crucial role that ICH-M7 guidelines play in protecting patients.
I'm glad to hear that. It's a fascinating and ever-evolving field.
It is.
And it's all driven by that ultimate goal of developing medications that are both effective and safe for the people who need them.
Absolutely. And I think you've done a fantastic job of highlighting that balance between scientific innovation and patient safety. So that brings us to the end of our deep dive today. But we hope you'll join us again next time as we explore another fascinating topic in the world of pharmaceuticals.
Thanks for listening.